Welcome to another episode of the Bioinformatics Chat. I'm Jacob Schreiber, and today I have with me Dr. Molly Gasperini. Dr. Gasperini got her PhD from the University of Washington working with Jason Dury, and now is a scientist working at Octant Bio. Thanks for coming on the show, Molly.
Thanks, Jacob. It's great to see you again and you and Roman virtually. And thanks for inviting me. It's been a while since I've talked enhancers for about a year and a half, and so I can't wait to do it again.
Yeah, I remember the last time we talked about this was at one of the ENCODE meeting back when you could still actually go outside.
That's right. I know. In the sunshine and in person. Yeah.
So you're a scientist working at Octant Bio now. Can you tell us a little bit about what Octant does and what it's like to work with them?
Yeah. Oh my gosh. Octant is a really fantastic team. It's a company founded by Sri Kasuri out of his UCLA tech dev and highly multiplex assay sort of based lab. They've been around for about 3 years, and they moved up to the Bay Area a year and a half ago when I joined. And the company is largely a tech dev company, I guess you could say, which is what really drew me to it. There's nothing to do with gene regulation, nothing to do with non-coding DNA, but it's largely focused on multiplexing assays of GPCRs, which are huge drug targets. And so the idea is if we can apply all this like multiplex tech stuff that uses like high throughput sequencing as the readout, stuff that uses like a lot of DNA barcoding, assays like deep mutational scanning, things like that. If we can pair that to genes and signal that is important in drug discovery and drug functioning, can we better understand drugs and design better ones? So it's a team of about 20 people. It's a very academic company. Thanks to Shri's influence, I just posted on the blog like some data that we had generated that we thought might be useful for the community. Last spring, we pivoted some of the tech at the company to a COVID testing and screening assay. So you can use next-gen sequencing to screen samples for COVID RNA much faster, SARS-CoV-2 RNA. And so it's a super fun place to work. And I highly recommend it to anybody who's interested.
So is the company pursuing its own research agenda, or is it contracted from other companies that want certain assays designed?
Its own research agenda, and there's sort of like 3 different groups, but we all really work interconnectedly, and it's largely and so like we have our own sort of biological goals that we're trying to hit, but towards that we're trying to build new biotech tools that are in this sort of like multiplexed realm that serve those goals. And then sort of from there, we'll like either hopefully have the company will have its own drug target or be able to have partnerships for other places that are interested in the tools we're building.
That sounds awesome. I think that it's a lot of your PhD or some of your PhD work seem to be involved in developing assays, these multiplexed assays. And so I think that's really interesting when people are able to take their PhD work directly and then continue doing something similar in the industrial realm after.
Yeah. And it's there's a fair amount of human genetics in the company as well, which is really exciting. There's not really any non-coding gene regulation, which is pretty funny. Which is why I was so excited to come and chat with you guys to see if I can still hack it as a gene regulation person.
Yeah, so let's delve into it then. That the reason we were interested in talking with you is because you've done a lot of really cool work involving enhancers. So in particular, the paper that I thought was super interesting and I've been recommending to everybody who wants to know about non-coding regulation is this Nature Review Genetics paper about basically seems to define what an enhancer is.
Or tries, yeah.
So with that simple question, what is an enhancer?
What is an enhancer? Yeah, it's so funny. I remember distinctly I was new to the Shinduri lab, a rotation student or something like whatever 6, 7 years ago that was now, and we had a journal club, and another grad student like threw his hands on the table and was like, "What is an enhancer?" And then certainly grad school, and then being part of the ANCO consortium and writing this with Jay and Jacob tome, not you, Jacob, sorry, but the in writing this, it's like there's been so much work. There's been so much amazing work in progress made in understanding this from this concept that was only really discovered in the early 80s, or first sort of demonstrated in the early 80s. It's 40 years of work, and it's I think kind of speaks to the complexity of human gene regulation. In terms of like how much is still vague, how much is still hard to pin down exactly what's going on, and how do we try and move forward in trying to understand non-coding DNA in the context of disease and everything like that, when it's still can be like a vague definition. So that's a non-answer for you. So we can delve into what I actually think an answer is.
Yeah, so maybe it might be helpful to first take a step back, which is that, this paper, actually, I think maybe like your thesis, which I was perusing while preparing for this.
How'd you find my thesis?
Just on your Google Scholar.
Oh, really?
The easiest way to prepare is just like open up all the papers that the person's published.
Oh gosh, I should have done the same so I can quote you back. Oh my goodness.
Well, so the gist is basically that each cell in the human body has the same genome, and there are trillions of cells in the human body that do very distinct things. How is it possible that the same set of genes can give rise to all these different types of cells? And how do they know, particularly given that we don't have, humans don't have way more genes than other organisms. We have a very modest number of genes, is that right?
I think it kind of depends. We have 20,000, right? And yes, some organisms that you wouldn't expect to have more, some have less. It's a fun thing to Google around. I don't remember any off the top of my head, but we certainly don't have the most DNA. We don't have the most genes. And I remember of the faculty in the Genome Sciences department who was sort of around when people were trying to determine how many, around like the late 90s, early thousands, how many genes do humans have? There was like a betting poll going around for like, do we have this many? Do we have this many? So it's, definitely not like we're the most complicated, the most fancy, so we have the most genes.
I remember Steven Salzberg giving a talk at ISMB in Chicago a few years ago.
Oh.
Where his keynote was basically like, how many genes do we have?
Exactly. And everyone's like, well, 20,000. He's like, well, yeah, that's awesome.
But exactly. He was talking about how like in the early thousands that people were like betting on the tens of thousands.
Right.
Like on that order of that. Now people are betting like within 1,000. And then obviously because it's, Steven Salzberg, that there's tons of drama involved in.
And controversy. And yeah, exactly.
Right. Okay. So then the reason I bring it up is because the way that gene expression is regulated in humans is not necessarily through just, the number of genes that we have, but through the combinatorial patterns of regulation through elements like enhancers.
Exactly. That's kind of how I tend to explain this. Okay, so we have about 20,000 genes, and we have way more cell types than that in the human body. But if you do the math or just think about all the combinations that you could get just from turning any one gene on or off, you have a vast diversity of possibilities. So it's all through the combination of if you can turn a gene off or turn a gene on, if all the combinatorial outcomes could give you a vast diversity of cell types. So
then you kind of gave a non-answer before. I guess the way that I was introduced to it is that in my undergrad, I took some biochem courses, and so I knew like what a gene was, and maybe the term enhancer was thrown around a little bit, like as like it's a thing.
Yes.
You need to know for a test.
Yeah.
And so I was mostly introduced to the term enhancer from the computational side as basically like it's a block in the genome that regulates gene activity. And so I never really thought of what was going on inside that block, just like it was a very distinct thing. And like people know, in the same way people know what a promoter is, which also isn't true. People
Totally.
know what an enhancer is. So do I get into what people talk about when they say enhancer?
It's funny the way you describe that, because I think that's sort of how the concept of an enhancer was first described in 1981 by Banerjee et al. They found a DNA sequence that did not code for a gene in the SV40 simian virus genome, very small genome. And they put this piece of DNA, and as you're saying, it was just like a thing, a chunk of DNA. They didn't really know what the sequence content was. They put it near a gene and far away from a gene on this sort of synthetic piece of DNA. So it's not in a human genome. It's just sort of, they're trying to express this gene on this other little piece of DNA, and they put it close to the gene, far away from the gene, and they found that, and they swapped the direction of this piece of DNA, of the viral piece of DNA, and they found that in all orientations, it could turn, it would just jack up the expression of that gene. And they're like, this is weird. Here's this piece of DNA. It's non-coding. It's not supposed to have, much purpose because it's not a gene. But it clearly is creating this effect of turning the expression of this gene up. And so from that definition, I think it was sort of like, okay, this is an enhancer. It enhances gene expression. It's a thing that's in the genome, a piece of DNA. I think there's many different definitions in terms of if you can distinguish what a promoter versus enhancer is. I think largely enhancers are supposed to act, they can act far away from the coding gene, whereas a promoter is supposed to be really close and immediately upstream. But there's, I think, whole reviews written about what a promoter is, et cetera. Around the same time when we put out our Nature Reviews Genetics paper, I remember there was one about what is a promoter, and it was like, this is excellent complementation. So that's sort of I think why that concept is sort of how it's viewed to some extent. But after that first one, there have been decades of work in finding individual ones at different places in the genome that regulate different genes in the human genome and mouse genomes and other organisms, fly, etc. From there, I think the picture kind of emerged as that these pieces of DNA largely hold transcription factor binding sites. And that's kind of a jargony term. But what it means is usually it's a short piece of DNA that allows a protein to bind to that piece of DNA. So it's a little bit like a signal saying, come over here, protein, and the protein can come, this enzyme can come and bind to the piece of DNA. So it's located somewhere now rather than just floating around in the milieu of the cell. And so vaguely, I think an enhancer is just a bunch of different sequence motifs, a bunch of different of those transcription factor binding sites. They have little codes. So like this transcription factor binding sites binds ATGC, ATGC. This transcription factor binding sites binds GT, 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 this sort of thing. And so I think enhancers can kind of loosely be seen at the sequence level from that. The only issue is that it's not so clear of a code. Like it's sometimes things bind where you wouldn't expect them to. They don't always bind. It's highly complicated. There's a lot of variation in what those little signals are, those little sequences are. There's other things you can kind of like look at and say, okay, so it's got all these transcription factor binding sites in the DNA. There's all these epigenetic signals, which is essentially like how the DNA is wound at that particular location. What other proteins are binding around that location, where the DNA is in the cell, and then there's more and more sort of characteristics like that are being added. So that's sort of like kind of like what you can imagine a enhancer is. But then the big issue is that you see all these descriptions. You see it's got these transcription factor sequence motifs. It's got these proteins there. The DNA is wound in this way. It's located near a gene such that you think it could be like it's located in 3D space near a gene, not necessarily in DNA linear space near a gene, so you think it could look like an enhancer, but then if you delete the sequence or if you turn the enhancer off using some other synthetic methods, the expression of the gene doesn't change at all. And then you're like, wait, but I thought that's what an enhancer was. Like I found one. And that's kind of the new thing that with a lot of new technologies such as CRISPR screens, which we describe in the paper, or other reporter assays or these other sort of high throughput techniques that that's kind of brought us forward to be like, wait, so is this what an enhancer is? Is it the technology that still needs to catch up? Like, are we not looking at it in the right way? Is it in the wrong cell type?
Yeah, so you're basically saying that like people had these definitions of what an enhancer is, that like, oh, an enhancer is something where these transcription factors bind to it. Transcription factors, the proteins that or related to transcription.
Yep. Yeah. That's right, exactly.
Or either the transcription factor binds here, or these other marks like the histone the histones near the enhancer get modified in very characteristic ways.
Yep.
And you have this really interesting comparison in the paper where you talk about the blind men and the elephant.
That's right. I forget how, I think Jay maybe mentioned that, or so, and we were like, should we go for it? And we're like, fine, this is a pretty silly, great metaphor, a classic one, I think, in biology. And I think it definitely applies here where, the people who work on reporter assays will say we should do this. The people who work on, understanding transcription factor binding site sequence motifs will say it's this. Yeah, and I think there's a lot of, I think, at least what the thesis we have in the review, and that I personally have the view that it's going to be a fair amount of layering on these techniques and actually trying to like read out and use sort of like classical genetic logic of like, if you can perturb the sequence, if you can get rid of the sequence, do you see the expected effect? And if you put the sequence back, do you see the rescue of that effect? That's kind of like how I'm envisioning an enhancer could be defined. But again, that's a genetics perspective rather than like what's the true biochemistry of what's going on at the locus, which is still a huge can of worms.
Right. The metaphor being that if you have if you ask blind men to describe an elephant, that they would go around, they'd like touch it, and they'd be able to provide details about what an elephant felt like or sounded like, but they wouldn't be able to describe what an elephant actually was.
Yeah. Someone touches the tail and is like, oh, it's like a piece of fur. And someone touches the ear and they're like, oh, it's like a leathery jacket or something. Yeah.
Right. And so you can get these similar descriptions for enhancers, like you mentioned before, transcription factors bind here, histones get off, but none of those describe what an enhancer actually is.
Right.
And so what you were trying to get to is what a definition of what is actually going on with the enhancer.
Right.
And so it's not just, you brought up this contradiction where if you define an enhancer as like it has to regulate the expression of a nearby gene, or you find some of these positions that seem to be enhancers based on transcription factor binding and histone modification, but don't actually regulate expression. So what do you think an enhancer is personally?
Oh gosh, which is definitely all controversial. But yeah, what Jay and I were putting forth, Jacob, were that if you can, if it looks like an enhancer, if it smells like an enhancer, if it's located where we think one is, or where it should be, etc. And then if you delete that sequence, like you take it out of like the cell type that you really think is where this enhancer should be active, based on other biological studies or whatever. If you delete that sequence, and then the target genes that you would expect, that's sort of relatively located nearby, if that target gene goes down, then like if the expression of that gene goes down, there's a good chance that the thing you deleted is probably an enhancer. And if you put it back in, then presumably it is also, that's just like for further validation. It's harder to do both pieces of logic in like large screens. The other issue with enhancers being that there's a huge amount of non-coding space to search for enhancers, and there's a huge amount of cell types in which an enhancer could be active or not. If you delete it in the wrong cell type, you're not going to see anything. But if you delete it in, say, this other cell type, like if it's an eye enhancer, you delete it in an eye cell, you're going to see it active. But if you delete it in a skin cell, you're going to see nothing. Like that's a whole other added complexity. So we were sort of saying if you can sort of profile it, so you have a select number of candidates, and then if you can delete it, ideally in a screen or just individually, then the chances are that you probably have a real enhancer. That's as far as I'll go. And it's a little bit often that we propose like, this is what we're going to define what an enhancer is, but instead we're sort of like, here's another way to just be like get more increasingly sure that like what you're finding is truly an enhancer. But part of the issue is just, you read about individual examples of enhancers, gene regulatory elements, and it's incredibly just complex. Each individual enhancer that papers are built around, just one or two or like a couple near each other. You come up with exceptions, you come up with different mechanisms for how they could be regulated and working. And so this was our attempt at trying to say like, okay, this is probably another piece of evidence that the tech is appropriate for now in 2020, and that and that I think will bring you closer to truly finding something that we could call an enhancer.
That makes sense. It seems like your definition of enhancer involves like a causal mechanism, that the enhancer has to cause a change in expression. Specifically, it has to cause an increase in expression in the nearby gene.
Right.
And so proving that causal link technically can be difficult.
Yes. Yeah.
I mean, that seems to make sense to me. I think one of the ways that I had like a mind blown moment when I was talking with Rick Myers. He used to be the chair of the Stanford Genetics Department and now leads Hudson Alpha.
Uh-huh.
I was talking with him and he was mentioning that after finishing the Human Genome Project, they knew what the identity of every nucleotide was. And so they're like, okay, well, now let's move on to the simpler problem of assigning a function to every nucleotide in the human genome.
Right? And here we are. Yeah.
Yes, it's been much longer and we have not made a huge amount of progress. But it really like blew my mind that his view of it was that you should go through instead of viewing like an enhancer and promoters as like boxes that sit on the genome, that you should view every nucleotide as either having some sort of effect and quantifying that effect or not having an effect.
Yeah.
And so when you talk
like
about
that.
how enhancers contain lots of like transcription factor binding sites, it's really just saying that there are lots of nucleotides that are functional within the like what you view as like an enhancer box.
Yeah, I would totally agree with that sort of thing. Yeah.
Yeah. And so I've always thought that that's particularly interesting because what you're talking about is how one of the complexities in defining enhancers is that enhancers seem to be cell type specific. And so, and if you're looking in the wrong cell type, you might not identify that there's, an enhancer, but really it's just part of the complexity of like, okay, this nucleotide enhances expression in, lung related cell types, but not in skin related cell types. I was mentioning this earlier, but I really like one of the first paragraph of this paper. You end it by saying like, however, despite the clear importance of human enhancers, blah, 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 there's a tremendous amount we still don't understand, including where they reside, how they work, and what genes they mediate their effect for.
Oh, gosh, because it's true. In a lot of ways, after 40 years of work, it's still like such a can of worms. But 40 years ago, 1981, when this concept was first described, it wasn't like, there was one from a virus, like we knew so little. So now, like, we know so much more. Yeah, it's like, part of it, I think, part of why that first paragraph is so caveated is that, is because, biology, nothing is certain. And when you try and like define, like 98% of the genome or screen 98% of the genome for these things, you can't be like, okay, we know for sure that we've like characterized each one. But like, it's not to say that there's not been a huge amount of work done, but it is, I think, more to say that like there's a huge amount of promise here in further understanding in some part of our DNA that might actually be like really, really important.
Right. Yeah, exactly. That it's like you mentioned that in the last 40 years, we've come so far, but and part of coming so far is realizing that the problem is so much bigger than we thought that it was going to be. Yes.
Yeah. I love that those perspectives from genome scientists who were around when the genome was first mapped, and then like, the next generation of can of worms.
So there's a huge table here where you talk about every single way that people have used to identify enhancers. Do you want to talk about each and every one of those ways in depth?
Just like my general exam, exactly.
No, but maybe maybe it'd be helpful. Like what are some fairly modern approaches that this is a massive problem. So how are we collecting data about it right now?
Yeah, there tend to be loosely three categories. How I think about it. There's what does the DNA sequence itself look like? So that's sort of looking for transcription factor binding sites. That's sort of looking for patterns in just the DNA sequence itself. There's loosely biochemical annotation, which is sort of saying, okay, it looks like the DNA is wound this way. It looks like the DNA is located here. It looks like the DNA is has these proteins attached to it, all the other sort of things in terms of like what it looks like it should be an enhancer. That's the second sort of category. And then the third one I tend to think of as the scaling up of this tech is relatively new. I tend to perturbation assays. So this is where you either turn an enhancer off, or you delete it, or you try and disrupt its function somehow. And that's sort of the third category. And in this third category is kind of why we were asked to write this review, in that it's relatively new in terms of being able to perturb enhancers or any piece of DNA at scale, meaning like in one experiment that will take you maybe, if all goes well, like 1 or 2 months, you can screen thousands of enhancers. You can screen tens of thousands of genes or things like that. And so these are the scaling up of perturbation screens largely is due to the advent of CRISPR as a perturbation genome disruption, genome engineering tool. It just makes it incredibly easy to delete a piece of DNA, change a piece of DNA. If you attach other enzymes to the CRISPR protein, then you get you can maybe mess with the function of an enhancer and turn an enhancer off, as a loose way to say it. And because you can wield CRISPR so well and so easily, and it's so well adapted to high throughput screens or these massively parallel multiplex assays, it's kind of a new advent in that, like, oh, we could actually maybe not just like delete one enhancer and study that for 10 years or 20 years and like create many papers about this one enhancer, but instead maybe we could try and tackle like maybe someday all of the stuff that has been nominated by assay type 1 and 2 by sequence understanding and biochemical annotation. So those are kind of like the three main categories of how we take a look at enhancers, I think, now. And the third one, the CRISPR screens, massively parallel reporter assays have kind of come to the forefront as ones where we can actually perturb the function of a piece of DNA at a scale that the size of the human genome requires.
Right. That scale must be massive. You mentioned that the non-coding part of the genome is massive compared to the coding part, and so having to check almost all of that is ridiculous.
Yeah. But luckily, that sequence space has been narrowed down because of all the biochemical annotation, because of all the sequence understanding.
So do you think that it's sufficient to use things like so like we were mentioning how there are these, there are these classic signs of an enhancer, transcription factor binding, histone marks, various types of things, but some of those end up not being enhancers. So do you think it's sufficient to limit your search space to those positions, or do you think that there are some enhancers that, or it's kind of like the looking under the street lamp phenomenon?
Street lamp, the other metaphor. Yeah. That's a hard one. Because I'm sure there are examples to every one of those pieces of evidence, where it's like not every enhancer maybe requires a, H3K27 acetylation, like maybe like all these other different types of biochemical annotation or sequence motifs. But I think it's a great starting point to narrow down the search space. And part of this is that, to say a sequence is not an enhancer is even harder than saying a sequence is, I think, because the fact that you have to check every possible cell type in the human body to possibly say, no, this, as Rick Myers sort of defined it, the sequence does not have function, because, DNA function changes, as we were saying earlier, across every cell type in the human body. That I think is almost harder to say. So the sequence is not an enhancer. But in terms of like, if it's sufficient to first check for, in, say you have one skin cell type and all the biochemical annotations point to it being an enhancer. The sequence annotation also does. I think is a great starting point, and then we can kind of see what comes from that. And try and scale that across many different cell types, see what patterns come from it, see if the CRISPR screening tech or whatever massively parallel reporter assay screening tech, like if that all You kind of we should it's still nascent, so we should kind of do a first pass, I think, with these uh, annotations leading the way, and then from there be like, okay, like, is this what we'd expect? Should we search in non-coding DNA that it doesn't look like, it doesn't have these correct characteristics of different biochemical annotations, and then see. So I think the chances are it's a great starting point, but there's most likely exceptions.
That makes sense. It seems like so you're mentioning that this primary tool are these like massively these like CRISPR assays, and these rely on single cell technologies, and so it just in order to scale up, that if you have an experiment with like 100,000 cells, 1 million cells, that directly connects to the number of positions that you can perturb. But to date, single cell experiments have mostly been somewhat small. Do you have it's small to the point where you're not even able to perturb all the positions that you might like these candidate. You might not even be able to perturb every single candidate position within a single cell type in a single experiment. Do you think that these types of single cell experiments are going to be able to scale up to the point where you might be able to look at every single candidate region in one experiment?
Towards single cell, I think the answer is yes. I think single cell screens eventually will hit the point, and they're coming close to having the scale and cost that I think would be possible. But not all CRISPR screens or massive parallel reporter assays require single cell experiments for sure. The first paper I published wasn't one. It was a non-coding screen based on the function of one gene, and I think there's like a bunch of different functional assays you can use in CRISPR screen that doesn't require single cell RNA-seq as your readout. Single cell RNA-seq is just great because the phenotype is literally the expression of all the genes in a cell, and that's literally the definition, at least that has been largely proposed for an enhancer in terms of impacting the expression of a gene. So there are many other different sort of CRISPR screens, but usually they're limited to checking the function of maybe a handful, maybe 10, 20, 30 genes. Because and that's like optimistically, usually it's less than 10, just because you have to kind of create a specific functional assay, or a more bespoke transcriptional assay for a handful of genes. Where single cell RNA-seq, you kind of get that all for every expressed gene. Okay, so towards your original question, I think single cell RNA-seq is definitely hitting the point where it's going to be more reasonable to get more to be able to attack more and more regulatory elements to actually meet the scale of what is required and has been nominated by biochemical annotations. I know like 10x Genomics is coming out with new ways for their single cell RNA-seq kits to kind of like super jack them up such that instead of getting maybe 8,000 cells, single cell transcriptomes out of a kit, you get 10, I don't want to misquote, but like 10 times more or 20 times more, like super load them essentially and have that be feasible. I know Jay's lab has been at the forefront of combinatorial indexing as a way to be able to run and get like 3 million cells, single cell profiles, and really scale up single cell ATAC-seq or single cell RNA-seq. Jay's lab, Xanash lab, Andrew Ady's lab, Georg Selig's lab. There's a ton that are sort of working this space to create sort of like at-home kits where people can use this, or not at home, but like non-biotech produced kits. And so I definitely think that stuff is coming around. But at the end of the day, single cell RNA-seq certainly requires a ton of sequencing, Illumina sequencing. No, you just can't get around it. Sure, you can make all the reagents and the kits and everything cheap, but you have to still sequence the heck out of the transcriptome. And I think that will always be a limiting cost, because for every cell you add more sequencing, and that's just highly expensive. So it's all very expensive right now. I think the tech is there, but I think the cost right now is what's really prohibiting this from being like widespread use.
Gotcha. So we basically need a whole new, we need a whole new grant infrastructure just for studying enhancers.
Oh God, maybe a whole new grant structure for many things, but yeah. I do have some optimism that stuff will be cheap enough, but eventually. But I think it will take a while, unfortunately. Yeah.
Again, coming at this from the computational perspective, I see tons of papers that come out in various journals where people are trying to predict enhancers. And so it's always interesting to me that there's so little agreement on what exactly an enhancer is that to try to like predict them seemed like a very ill-posed task from a computational perspective. And after reading works like this, I've become super skeptical of any type of computational method that doesn't come from a collaboration with a wet lab.
Interesting. Do you think part of that is because there's not enough data so far where people are pretty confident that they found like a functional enhancer?
I think that it's a combination of two things. I think it's definitely that there's not enough high quality data doing things like promoter enhancer interactions. And I think the second thing is that a lot of computational people don't really care about the subtlety, that they're looking just to get a paper out. And so they're like, I made a deep learning method that targeted this thing. And I download these annotations from the internet.
Yeah.
They'll like go to Phantom 5 and they'll download like permissive enhancers, and they won't know what a permissive enhancer is. They'll just say that it's important to identify enhancers, which it is, but then they won't really care about the label set too much.
Interesting. Okay. And that's largely what your podcast is sort of trying to address, right? Is trying to like fill in some of that for people who are like, I have a computer science degree. I'm not going to dive into, PhD in gene regulation, but like trying to like explain the depths a little bit more of the complexities, a little bit more of biology.
Sure. Yeah. I mean, I would definitely encourage computational people to look very deeply in, because there's so many errors that you can make from the computational perspective if you don't really understand what's going on with your data.
Yeah.
So I think with the time remaining that we can move on to the simple question of like, what's actually going on in an enhancer? Like we've been able to define an enhancer kind of in terms of like, maybe there are some marks, maybe there's some like, the important sequences there. We know that we've identified an enhancer when it messes with gene expression, but how does it actually influence gene expression?
Yeah, there's a running joke at work at Octant, and I came from Jay's lab where Jay has doubled down on like I'm a technologist, like the lab is sort of driving towards like a biological vision, but largely, it's a tech dev place. And before I was in Jay's lab, I was in human genetics labs, which people can also argue aren't biology labs, and so at Octant, the joke is like, everyone's like, ask biology questions. And I'm like, I'm a tech dev person. Like, like don't ask me to like make big biological claims or, that sort of thing. But I'll give a crack at it. Largely from education I got from University of Washington Genome Science PhD program, a class by John Stamatianopoulos, things like that, reading papers like that, learning about the history of these things. But I'm definitely not an expert in the literature in terms of like the latest like concept and diagram. Like I was worried about some of the diagrams we have in this Nature Reviews genetics paper because they're relatively simplistic in terms of like what people actually truly think is the mechanism of what's going on in a enhancer. But here's sort of how I understand it, is that you have a cell, and again, that cell has the same DNA sequence in it as every possible or not every, but like largely the same DNA in it as every cell in your body. However, in that, what makes that cell unique and what makes it a specific skin cell rather than an eye cell or a lung cell, for example, is that there are, again, these different proteins, enzymes that are expressed. And so how an enhancer plays a role in that is that you have DNA sequence when other transcription factors, enzymes that can turn genes on, this DNA sequence is present. The DNA sequence is there at the right time. Other like epigenomic or like other marks have like created a situation in which this DNA is available and accessible and wound in such a way that like these other enzymes can access it. It has somehow potentially been moved in space, in the 3D space of the nucleus in the cell, to be potentially close to, like in actual physical space, to its target genes or target gene region. Maybe it's just this one enhancer acting by itself. Maybe it's a small network of enhancers or gene regulatory elements. And essentially, because this DNA sequence has certain transcription factor binding sites and it looks a certain way and it's got these other marks on it that are sort of accumulated over the cell's lifespan, it can flag, hey, RNA polymerase, hey, like, come over here, and then somehow enrich this little tiny environment for increasing the chance that RNA polymerase is going to come by and transcribe whatever's in that region, most likely, most weighted towards transcribing the actual gene that is supposed to be turned up to be enhanced, the expression of which is supposed to be enhanced. And there's, I think, a huge body of literature, of which I'm no expert in terms of like how exactly this region is, what's happening biochemically there, in terms of how this increases the chance that RNA polymerase will come and start and go. How, there are many different like ways, all the sort of transcriptional machinery, all the enzymes that control transcription, many different theories about how those are regulated, etc. And but yeah, largely that's my understanding, is that it's sort of increasing the chance that RNA polymerase, which is just being expressed elsewhere and just sort of like floating around, is going to be brought to this region, stay at this region, and transcribe DNA into RNA at this region. It's general enough that I can get away with it, but yeah, maybe that could be your next podcast in terms of like the actual biochemistry of what's going on. Yeah.
I mean, it seems kind of like by definition, I didn't really think about this until somewhat recently, but if you're saying that if you're saying the gene expression is going up, you're kind of by definition saying that RNA polymerase is transcribing transcribing the gene more. And so kind of, it follows by definition that an enhancer somehow has to increase the rate at which RNA polymerase is recruited.
Yeah. And plus all the stuff that creates the transcriptional. Yeah, exactly. It's not just the one enzyme. It's a whole suite of enzymes that make that happen.
I remember the same ENCODE meeting was the last time we, talked, that there was this like secret enhancer meeting where like Erez Lieberman Aiden wanted to have a like, he wanted to just like talk about enhancers with some people. And so through word of mouth, he was like going around talking with people. And I think people were trying to find you, and were you there?
I don't remember talking. No, with Erez. No. Yeah.
I think that people were trying to find all the people who were interested in enhancers, and we met late one night after the poster session. Yeah, it was super poorly planned, and just hearing the people that give ideas for how this was working. Like, one of the ideas was basically that like the transcription factors basically form like this big like finger, and what it does is like it pokes an RNA polymerase that's stalled at the promoter of the gene. And so that wouldn't involve some like complicated biological mechanism. It basically just like help dislodge a polymerase that had been stuck.
Like physical. Yeah, exactly. Yeah. Is it something that just like physically moves it? Is it something that changes like the pH of that region? Like there could be a large number of reasons.
Exactly. Right. Yeah. And so the whole, it really, it again opened, every time I learn a little bit of biology, I describe it as like, it's opening my eyes.
That's wonderful. That's great.
Just like you were saying, that there's so many different chemical steps that are there to build the polymerase machinery at the promoter and then have it go. And each one of those has a like association, dissociation constant associated with it. And one of those has to be the limiting factor. And so it seems like an enhancer is acting on one of those chemical steps in the process of building, of assembling and setting the polymerase, setting the polymerase there. So one of the things I found really interesting, again, I have no evidence of this because I'm not a biologist, but one of the things I thought was really interesting was that you can have an enhancer that is acting on one of the steps. It is increasing the association-dissociation ratio in the process of building the polymerase machinery, but that's not the limiting step. And so if that's not the limiting step, something else happens to be the limiting step, that enhancer, even though it's still active, would not show any enhancing activity. Yeah. So there's a number of reasons why something might not act like an enhancer, even though it looks like one. So it's even harder to say, how is this truly not an enhancer? Cell type, like perturbation method, like, yeah, I would totally agree with that. But if you do get that signal where you disrupt the enhancer's function somehow and you see the decrease in expression, then it's much easier to define it that way in the way I logic through it, at least. Yeah. Right. Of course. I think my favorite suggestion from that meeting though was that you create like a transcription canon, which is that the enhancer like basically helps form the RNA polymerase machine. Yeah, loads up and then shoots it at the gene.
That's great. That's funny.
I don't think that that's the right answer.
The biochemists might have a different opinion. Yeah, exactly. That is one thing I always felt in, feel in genomics and human genetics that, I really wish I had enough biochemistry friends to really be able to understand, a lot of those things. I always felt that was missing in some of my education in gene regulation and my understanding. But I think that's
one of the issues right now is that there's just so much stuff that, you can be a computational person and it can be a full-time job just keeping up with the computational literature.
Exactly. It's highly complicated and niche. Yeah.
And then someone's like, oh, enhancers are hard. You're like, I don't have time for that. Just enhancers are whatever I download from the internet.
I know. That's I think it's part of the genius and also the challenge of academia or what have you, or any sort of science field where like there's so much power in being such an expert in your tiny niche, but then there's also, everyone is so myopic because there's just so much learning and understanding that it's really hard to kind of be able to even connect with other parts of science. Yeah.
Yeah.
So it's really cool that you're doing this podcast to try and do just that. Yeah.
Yeah. Thanks. I think that we're kind of coming up on the end of this. Is there anything else you want to say about enhancers before we close?
Oh gosh. I'm sure I've said some things that are debatable and wrong. Hopefully some things that are right in this. I had a great time chatting about it with you, Jacob. But, people listening, thanks for bearing with my open mind and naive understanding of gene regulation. But I'm happy to engage with people further as far as my understanding. But yeah, there's so many experts that I'm sure you're sort of going through in your podcast. I really appreciate you inviting me to give my naive perspective.
Yeah, thanks for joining us, and we'll be sure to provide your contact info so that people can write angry emails about how they disagree with you.
You can put my Twitter profile and make sure it's all public. So that's too funny.
All right. Well, thank you again.
Yeah, thank you for the invitation. Great. I had a ton of fun.